Welcome to the Invasive Mussel Collaborative webinar, Developing Innovative Control Technologies. Thank you for your patience this morning as we had to resolve some technical difficulties with the audio. Everyone should now be able to either dial in with their telephone or use their computer to hear the audio. And again, thank you for your patience as we were a bit delayed due to those technical difficulties here this morning. This is Erica Jensen with the Great Lakes Commission, and on behalf of the Invasive Mussel Collaborative, I'd like to thank you for participating in our webinar today. For those of you who are not familiar with the Invasive Mussel Collaborative, we are a collaborative identifying opportunities to advance scientifically sound technology for control of invasive Dreisseniid mussels in order to produce measurable ecological and economic benefits. It's widely recognized that invasive zebra and quagga mussels are causing significant impacts across North America, and numerous efforts are underway to prevent new introductions, eradicate newly discovered populations, and mitigate the negative impacts of mussel infestations. These efforts are ongoing, and in some cases, more work is needed to enhance our understanding and ability to manage and control these invasive species. The Invasive Mussel Collaborative helps in this way to provide a framework for communication and coordination, and is working to identify the needs and objectives of resource managers, prioritize supporting science, and align science and management goals into a common agenda. If you'd like to know more about the Invasive Mussel Collaborative, we encourage you to visit our website at www.invasivemusselcollaborative.net. The website is intended to serve as a central hub of information on Dreisseniid mussels. It also includes background information on the collaborative and recordings of past webinars. If you know of information or have resources that you would like to see added to this website, please contact us. We also encourage you to join our Invasive Mussel listserv if you have not done so already, to ensure that you receive future webinar announcements, get information about recent Dreisseniid-related news, and have the ability to connect with other researchers and managers working on zebra and quagga mussels. A link to join the listserv is provided on the collaborative website. Today's webinar is part of an ongoing series of webinars hosted by the Invasive Mussel Collaborative. The purpose of the webinars is to facilitate learning and information sharing on topics of common interest. It will be recorded and archived along with past and future webinars on the Invasive Mussel Collaborative website. Today, we're pleased to have representatives from the U.S. Reclamation, the U.S. Geological Survey, and the co-founder of BioBullets to share their experiences with developing Dreisseniid mussel management and control techniques, including methods and results, and answer questions about their efforts. In just a few minutes, we will begin our first presentation. But before we begin, I would like to go over a few logistical items. As I mentioned, today's webinar will be recorded and posted on the Invasive Mussel Collaborative website within a few days. We are pleased to have a number of people registered and participating in this webinar, and due to the large number of participants, we have set all attendees' lines in listen-only or mute mode. Attendees will remain muted throughout the webinar, and we will be taking written questions for our presenters. Thus, if you have a question, you may type it into the questions pane of your GoToWebinar dashboard. Questions may be submitted at any time during the webinar. Depending on the time, we may answer one or two questions following each presentation, but otherwise we will hold the majority of questions to address after all three presentations have been given. Again, you can submit questions at any time during the webinar. We will do our best to take all questions or comments during the webinar. If you have to leave the webinar before we have gotten to your question, we encourage you to review the webinar recording that will be posted on our website to hear your question and the answer at a later time. If you are experiencing technical difficulties, you may send a chat directly to the Great Lakes Commission organizer attendee. If you have technical difficulties, please send a chat message to the organizer. So with that, I will introduce our first presenter of the morning, Denise Hossler. As a University of Colorado graduate, Denise Hossler joined the Reclamation Department of Reclamation invasive species team in 1999. Working for the Ecological Applications and Research Group, she helped develop an integrated pest management manual and participated in integrated pest management research to control several aquatic invasive species, such as giant sylvinia, salt cedar, and Dracaenid mussels. Since 2006, Denise has worked on the early detection and potential control methods for Dracaenid mussels in the western U.S. She is currently serving as the Mussel Research Coordinator for the Reclamation Research and Development Office, assisting with the advancement of innovative technologies for the management of Dracaenid mussels in reclamation facilities. And then with that, it looks like we have your presentation up, Denise.
Oh,
so
great.
I will hand it to you.
Do you have my voice?
Yes, we can hear
Hi,
you.
Erica. Oh, great. Okay, so I'm going to discuss the mussel program here at the Bureau of Reclamation. I'll start off, for those of you who don't know, the core mission of the Bureau is the delivery of water in the western United States and provide hydropower. We deliver approximately 10 trillion gallons of water to more than 31 million people each year, and we are the second largest producer of hydroelectric power in the United States. With that comes a great deal of infrastructure. This just kind of lines out the miles and miles of variety of infrastructure. And all of these various systems and equipment are vulnerable to the effects of invasive species. As we know, invasive species are costly, and the technology is ever growing. We have a pretty well-developed invasive species program thanks to Fred Nibbling, who came on board and actually helped to foster this integrated pest management program here, utilizing mechanical biocontrol, chemical control, and environmental controls. Currently, these are the aquatic invasive species of concern, and of course today we're going to talk about zebra and quagga mussels, which impact reclamation facilities greatly. The initial findings were at Lake Mead in January of 2007, and in October of 2007, 67 miles downstream at Davis Dam, facility workers tried to pull a bulkhead gate out of the forebay, and the motor could not pull it up. It turned out that it was totally infested, blocking the ports for drainage, so they had to get a crane to pull it out. That then triggered immediate concerns on the management side because of the potential shutdown for water delivery and hydropower generation. Our structures were clearly at risk. The facilities were not built with redundancy, therefore the mussels would have a greater impact on our functions. As we know, the larval mussels disperse such large numbers. So it began a fairly large program that was geared towards being proactive and preventative measures, reactive, and retrofitting the systems. The initial program was, one, to come in and do facility vulnerability assessments, to initiate control research, and to do Dracaenid detection and monitoring. In those early years, we were doing about $4.5 million a year for this type of work. The facility vulnerability provides information on the structures within a facility that would be vulnerable, and it helps the facility managers with the federal budgeting. As we know, these mussels create the flow restriction issues, and that then impacts our ability to generate power and move water, these animals can live in a fairly high velocity system. Additionally, we are aware of the ecological impacts. These are some photographs of some of our facilities and the impacts. The primary issue that we see in our facilities is actually the shell debris. This is a water intake that's about 30 meters down. So the mussels obviously were quite successful in this particular environment. One of the extra impacts that we did not foresee was the enhanced aquatic weed growth. There in the Lake Havasu system, where the photosynthetic depth increased, the volume of aquatic weeds was very, very high. The big emphasis was to determine where in the West these organisms were. So this laboratory became highly funded to do that research. Between 2000 and 2015, we've looked at 425 water bodies in the West and almost 16,000 samples. 15 states collaborated in this initially, and in 2011 we began performing tests, all of the tests, both cross-polarized light microscopy and PCR on any sample where a body had been found. And this just shows you the spread through time. And I just wanted to point out that we have a few locations that are yet not on the USGS map, but our testing indicates that we do have some positive locations in those states. In 2009 and 2011, Fish and Wildlife fostered a round-robin testing with several laboratories. We also participated in this. What I wanted to point out is that the false negatives were the most common error for all methods. And because we were getting some positives in location where other laboratories were not, we began a vigorous research program. And basically, we determined a number of interesting factors, and probably the number one thing is that the buffering capacity of water is variable. Surface water is variable, and the shells will dissolve unless we buffer those samples at the time of collection. You can see that after 7 days, it's pretty difficult to see the villager under the microscope. This just gives you a larger viewpoint. We also worked on optimization of the PCR, and as you can see, these samples contain quite a bit of DNA, and getting these samples cleaned up proved to be quite a challenge. Initially, just tweaking all of the reagents in the tests, and I throw these up so that you can see that someone who is using 3 micromoles of milligrams or magnesium are going to have much different results. That's one of the fine-tuning that we did. Another piece was determining the proper prep kit. So you can see down at the soil kits, we had much better results. One of the interesting things is the preservation, the use of alcohol. We determined that isopropyl alcohol was adequate for the preservation of these samples, which is quite handy when you're out in a rural area collecting water samples. This graph shows you where the diminishment of the cross-polarized light under microscopy signal occurs, and where we begin to not be able to see them under the light, but yet our PCR will continue to tell us that there is DNA present. We are now using DNA assays for a number of different species. And I just wanted to give you a few things on our program. In the early days, we had 327 water samples and analyzed the positives that occurred and determined that 59.3% occurred at a boat launch or a marina. After doing 12,000 samples, again, our first-time positives do occur at a boat launch or a marina. And even for our ongoing positives, almost 50% of all of our positives are occurring at boat launches or marinas. Additionally, we have a number of one-time positives that we don't have ongoing positives. We're not entirely sure of all the variables, though we have a large database that we are going to be analyzing that information. And here is the value of the eDNA without the body. As you can see in this Arizona reservoir, we had some indication that they were present, but by the time we get up to about 20 villagers per sample, you can see that we've had indication of presence for some time. Again, this is a Utah reservoir, very similar kind of a setting. These are DNA results, and we don't actually see high numbers of villagers for some time. Again, this is a Colorado reservoir where we see the number of villagers under the microscope declining, and the DNA as well. Another New Mexico reservoir. Reclamation spent about $9 million on, this was American Resource and Recovery Act, and other additional funding for invasive mussel research. It has been, control research has been a large part of that research. We've done quite a bit of work with coatings and filtration in our facilities. One of the other things that we have done is a bit of UV testing. UV has proven to be quite helpful for our cooling water systems. We also are doing some turbulence testing. We have a young man that's doing some turbulence testing. We've done some improvements with fish screens using some of these foul release coatings, and they've proven to be quite effective on the fish screens, which is a real helpful thing because reclamation does spend a great deal of effort trying to preserve the native fishes out here. We have a number of other activities that are going on, including some more genetic work, trying to understand genetic differences in hopes of being able to find some vulnerabilities for integrated pest management. We also currently have some pulse pressure more oriented towards laser treatments. And then also looking more closely at the impacts and the costs. We're doing quite a bit of literature research and making that database available. And we have a website that you may visit at any time for current information. It's updated pretty regularly. And with that, that concludes what I have to say. So I thank you for having me today and hope that this has been informative.
Thank you, Denise.
I don't
see any questions yet. So
okay.
I think we are going to move on to our next presenter, and then we'll come back to you at the end with questions that come in the interim.
Sounds good.
Thank you, Denise.
Thank you.
So our next presenter is Jim Loma. Jim Loma is a research fish biologist at the U.S. Geological Survey's Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin. Jim's research focus for the past 6 years has been on building a research program to develop and evaluate Dreissenid mussel control tools for use in integrated pest management programs. Much of this time during this period has included the evaluation of Zequinox for open water applications, which has included laboratory and field evaluations to target and not target animals. Previous to his current position, Jim worked at 2 federal fish hatcheries, rearing a variety of cold, cool, and warm water fish and native mussels. He managed the fish culture facilities at UMESC, and he performed technical assistance work for the U.S. Fish and Wildlife Service's sea lamprey control program. So with that, Jim, I believe your presentation is up, and I will turn it over to you.
Sounds good. Thank you, Erica. So thank you, and good morning or afternoon, wherever you're sitting. Today I'll discuss our work in the development of a control tool to target filter feeding invasive species. Just to make sure everybody's aware, I'm presenting this webinar for John Amberg, who is the project lead for this effort, although I participated in some of the studies and I'm familiar with the work, so I'll attempt to answer any questions that you may have regarding it. There we go. Okay. So at UMass, we are taking a two-pronged approach to developing new control tools for aquatic invasive organisms. We're getting a lot of feedback here, Eric. I don't know if you noticed that.
If somebody is shuffling papers near the phone, if you could put yourself on mute for the time being, that would be helpful. Thank you.
I think we got it. Thanks, Laura. Anyway, so we're taking a two-pronged approach to developing the control tools for aquatic invasive species. We're trying to focus this work to minimize the impacts on native organisms while maximizing the impact on invasive species. Before discussing the delivery mechanism, I'll update you a little bit on the chemistry of it. So one of the goals is to identify new control chemicals that are not discovered yet and add those to our toolbox. In order to expedite this process, we developed these structure-activity relationship models, and we use those to predict the toxicity of those models, of the chemicals, and then screen large chemical databases that are out there in industry. For example, we screened the zinc chemical database, which contains millions of chemicals, and they found 30 potential chemicals that might have selectivity to cyprinids. We used the in vitro cellular assays to screen through these chemicals, and this did require us to develop cell lines for the big-headed carps, but several native species already have established cell lines. We have also initiated the development of the cell lines for zebra mussels. Once a chemical passes through this cytotoxicity trial, we'll then move it into in vitro testing or in vivo testing. If all goes well and shows promise as a control, we'll then begin the registration process. So if I didn't previously mention, this work was basically that I'm describing as on the Asian carps, but it's directly applicable to the Jacintid mussels. So the goal for this project is to create a delivery mechanism to take advantage of the filter feeding strategy of these invasive species. This requires knowledge of both the target animals and the non-target animals you don't want to impact. Our focus is, again, has been largely on the silver and bighead carp, but directly applicable to the developing particles for Jacintid mussels. So, again, the goal is to develop this oral delivery formulation that can selectively deliver agents that aren't necessarily selective in nature, such as anamycin and niclosamide, to control these filter-feeding aquatic species. There's 5 different basic requirements that need to be met for an oral delivery tool to be effective. First, it must be scalable for mass production. Secondly, it must be able to hold a toxin until the desired release time. Third, it needs to be the right size for the target animal to adjust. And fourthly, it must release the toxin payload at the right time, i.e. after ingestion. And lastly, of course, it must be readily consumed by the target animal. So as just mentioned, the technology must be able to be scalable so it can be produced in large quantities and also be affordable for production. One method for creating particles is microencapsulation. This is a process that is already being used to produce very large amounts of particles for several industrial applications. One method of microencapsulation is spray amortization. Spray amortization allows us to control the size of the particle by adjusting the nozzle orifice size and also adjusting the pressure. We selected this method for production because it is both cost-effective and it's flexible. We can use it to develop particles for carp, and then we can also tweak the method and the settings and use it to develop particles for Jacintid mussels. Secondly, for a toxic-laden particle to be successful, it needs to hold the toxicant for release at the right time, which in our case is after ingestion. We tested a wide range of particles that we created, all of which did very well in the leaching test. The worst of these formulations had less than 1.5% of the control agent, in this case anamycin, come out of it when it was placed into water. Another requirement for the oral delivery tool to be effective, it needs to be the right size so the target animals will be able to ingest it. So it needs to be size specific. For the big-headed carps, the preferred window seems to be between 50 and 100 microns. Native filter-feeding fishes like paddlefish and big-mouth buffalo, which are shown up here, prefer larger items, and native mussels prefer smaller items. So our technology needs to be able to manufacture particles in this 50 to 100 micron range, which is shown by these red bars. And it needs to be flexible, adaptable to create particles of different sizes. Spray amortization can easily create particles of this size and much smaller particles, such as those that would be manufactured for the dracaenids. If successful for delivering this particle, i.e. the fish actually or mussels actually consume it, then we need to make sure that it will release its payload under the conditions that we desire, which is after ingestion. So we looked at the presence of digestive enzymes in the gut of the carps. We found that there are some enzymes that are present at much higher levels in the carp than in native fish, and they're also present at very high levels early in the year when natives are not feeding. This helped us determine what types of materials we would likely need to incorporate into the particle to allow it to release after ingestion. And it also demonstrated that there are times of year when silver carp are actively feeding and native fish are not feeding. So there might be also a seasonality component that we could target for the carp. Then it comes down to the particles must be readily consumed by the target animals in sufficient quantities to be lethal. In order to determine how the fish would consume the particles, we conducted laboratory trials where we mixed largemouth bass with both bighead and silver carp. The fish were exposed to 4 doses of particles. They either contained 0, 10, 20, or 40 milligrams of anamycin. The doses were conducted in triplicate, and we repeated the study 3 times. The LC50 shown at the bottom of the screen indicate that largemouth bass are the most sensitive to the anamycin. So if exposed to anamycin that leaks into the water, i.e. they would take it up through the gills, they would most likely be the first animals to die. But as you can see, when we expose them up to and including a dose of 20 milligrams of anamycin, all of the big-headed carps died, but none of the largemouth bass died. This study was then ramped up to evaluate it on a larger scale. Trials were conducted in a quarter-acre pond. The pond was split in half. Each side of the pond had 90 fish, 40 big-head carp and 40 silver carp, plus 10 largemouth bass. We provided a particle into an area where they typically congregate to feed on one side. Then we counted how many fish died during the next 5 days. In this trial, we observed 50% mortality of the silvers and bigheads on the side that we dosed. None of the largemouth bass died. There were a couple turtles that were present during the course of this study, and one was even eating a dead carp, and they both survived. None of the fish on the control side died. This study is scheduled to be repeated in early July, and then they're looking to scale up into field trial applications. We are in the beginning stages of moving forward on and developing this technology for dracaenids. We can use the carp work that has already been done as a roadmap as we develop this technology. As for the particle, we can make it smaller by changing the orifice size and adjusting the pressure. Initially, we are looking at incorporating the closamide, which is already a registered molluscicidal compound. It's also used in the sea lamprey control program, so there is some registration history there. That's why we have chosen that particular chemical. We'll also have to do some formulation work to make sure that we get the buoyancy correct. For dracaenid particles, we know we can scale the production appropriately. We know we'll be able to hold the toxicant, We still need to confirm particle size, assure the particle will release its payload upon ingestion, and make sure Geosinids will readily consume them. Previously, we conducted a filtration study that compared the filtration of large and small zebra mussels with 2 native uniotid mussel species. We looked at the removal of the particles ranging from 1 to 10 micron in size. When standardized to tissue dry weight, zebra mussels consistently removed far more particles across the entire size range than native mussels did. This is promising because we likely have a window where we can apply a dose that will kill Geosinids and not kill native mussels. When we looked at digestion, similar to what we did with the Asian carp, we found that there wasn't any enzyme that was more prominent in the zebra mussels than in the natives. However, when we looked at how those animals are functioning at colder water temperatures, we see that some enzymes in the zebra mussels increased, which is shown in the green bars on the right side of the graph. This figure shows the change in enzymes between 24 and 18 degrees Celsius, respectively, which were summer and fall measurements. As you can see, the natives all decreased in enzyme activity, while the zebra mussels either continued or increased digestive activity. This might mean that we could treat later in the year, similar to the Asian carps, and thereby decrease the susceptibility to native mussels. So in the future, we need to make sure our nicosamide particles are the right size and the buoyancy prior to testing. We'll then conduct efficacy trials in a lab and then scale up testing to include trials in our bioassay trailer and hopefully into field trials. So to summarize, we're looking to control filter-feeding aquatic invasive species in 2 ways. We're looking to identify and find new chemicals or agents to selectively control them, and then to engineer new ways to deliver those controls that will make them even more selective. We have been able to develop an orally delivered formulation that was successful in killing big-headed carps in the laboratory and pond trials. And this will be the same approach that we used to develop a control tool for the Geosinid mussels. A benefit from in developing the Geosinid particles is that we've already been and continuing to learn new things in the development of the carp particles that we won't have to relearn. So with that, I'll take any questions that you might have now or after we're done.
Thank you, Jim. I think we are going to go ahead and move right along to our next and final presenter, and then we will go through the questions at the end. So our next presenter is David Aldridge. Dr. Aldridge is a professor at the University of Cambridge in the UK, where he heads the Aquatic Ecology Group. He is also the co-founder and managing director of BioBullets Limited. The BioBullet is a patented technology for the control of invasive filter feeders such as zebra mussels, quagga mussels, and Asian clams. The product is environmentally friendly, has regulatory approvals for use in UK drinking waters, and is a solution that has been used successfully by many UK water utilities, irrigation systems in Spain, and aquaculture facilities in South America. The company is looking for opportunities to demonstrate their products in North America with an aim of establishing partnerships and service providers.
Hello, thank you very much. Can I just confirm that you have my opening slide on?
It looks like we are viewing your other screen, not your presentation screen.
Right. That's probably not a good thing.
There we go.
Is that better?
Yes. Perfect.
Yeah? Okay. Excellent. Thank you very much for inviting me along. So I hope to present to you today a technology which is already applied and very effective within European freshwaters to control invasive mussels. So I'm going to structure my talk around 8 key questions which I anticipate people wanting to ask. So the first question I want to ask is, what is the BioBullet and why haven't I heard of it before? Well, the BioBullet is a technology developed by two Cambridge University professors. I'm an aquatic ecologist. I head the university's aquatic ecology group, and I'm interested in mussel biology, conservation in freshwater ecosystems, and environmental management. And my co-founder is Jeff Mogridge, who is a chemical engineer who's an expert in particle encapsulation technologies. And our idea has won many national and international awards. We've won the UK Research Bioscience Prize. We've won the NTEC Medal for the biggest contributions to environmental protection in 2007. And we have global patents protecting our concepts and technology, and that includes patents in Canada and the US, which gives us rights on producing microencapsulated products for controlling filter feeding organisms. We have a dedicated manufacturing plant in the UK with a capacity for producing 3,000 tons of our products per year, so we're already at a fully commercial scale. So are mussels that we're experiencing in UK as bad as those problems you're having over there in North America? Well, the answer very firmly is yes. We share similar problems, so we're confident our solutions will work over there. This image here gives an indication of one UK raw water from a water utility. In that pipeline, which is about 3 miles long, over 1,000 tons of mussel biomass is removed every 5 years. So we have big problems. And then we are very familiar with the problems associated with the removal and disposal of that waste biomass. So how does the BioBullets work? Well, typically when people try to control zebra mussels and quagga mussels, they try a range of things. Physical removal has been tried, sound waves have been tried, people try giving them electric shocks. There's a Mexican company that's tried chili pepper extract, but by far the most widespread and licensed technique is the use of chlorine. The problem with chlorine is that mussels are really very clever, and they can taste the chlorine in the water, and they close their shells. And they can then stay closed for about 2 to 3 weeks while the chlorine passes them by, so they can protect themselves from exposure to standard toxicants in the water column. So what we do with the BioBullets is very simple and effective, and that's what this schematic here summarizes. So if we start at the upper diagram labeled A, then if we expose a zebra or quagga mussel to a toxin, they'll taste that in the water and they'll stay closed for 3 weeks. What we do with the BioBullet is that we take an active ingredient which is toxic to the mussels, and we encapsulate it in a tasty coating which is just the right size and shape for them to filter from the water. And so they swallow our poison pills without realizing that they're taking in a toxicant. And a single mussel will filter about 2 liters of water a day, and so they die very, very quickly. What's more, we engineer our products so that anything which goes in the water that the mussels don't filter out will dissolve to harmless concentrations very quickly, within hours, and therefore there's no accumulation of our products in the environment. And so it's very safe in open water conditions, in recipient water bodies. So here are some images under a scanning electron microscope of some of our particles. In this case, we've put potassium chloride inside the particles, but we can put anything in within our patent. So you can see they're beautifully spherical and just the right shape and size for the mussels to feed on. Here we've freeze fractured one of our particles, and you can see in this case the salt crystal which is going to give them their toxic payload is here, and it's encapsulated in this tasty coating material. But as I say, we can put any active ingredient inside our particles, and indeed that's what we do now. So do we have any case studies to show you? we've got many case studies. I'll give you a couple in this presentation, but the product is widely used now across the UK water industry. So the first is water utility Eastern England. We dosed as an end of season dose one of our formulations called SB1000 that has a different active ingredient than the potassium chloride I've spoken about before. And we did an experimental trial in the draw-off tower in the reservoir where we dosed into one pipe, shown on the right here, and we also had a control pipe going onto the works as well. So we had an experimental setup within the pipelines, and we monitored the mussels coming through both pipes. And so on this figure here, we've got a 6-week period along the bottom of the axis here against the percentage of gaping mussels, so those that are freshly dead and with their valves gaping is shown in the photograph. And what we can see is during this 5-day dosing period here, we started to get mortality in the dosed pipe, going up to about 60% of the mussels coming through that were dead, compared with a much lower proportion, less than 5% at any time, coming through the control pipe. So an effective kill. The second case study I want to show you gives a slightly more quantitative impact on the success of the product in an operational waterworks. Again, this was a reservoir-fed water utility. Water is fed from the reservoir shown in this image down a 400-foot long, 3-foot diameter pipe, and mussel fouling in that pipe had reduced the capacity of the pipe to 56% of its design maximum. We dosed our SB1000 product for 10 days continuously at the end of season, and the capacity of that pipe increased to 94% of its design maximum, and that was evidenced by a large biomass of dead mussels coming out of the pipe, 650 cubic feet, from a relatively narrow and small pipe. So again, very effective. And the product has now been used very successfully by 4 different UK water utilities, the largest utilities here. Does it harm non-target organisms? Obviously, that's a big concern. Well, as I said before, we engineer our products so that they break down within approximately 3 hours of going into the water. So anything the mussels don't filter will dissolve to harmless concentrations, so there's no impact on non-target organisms. And we have the capacity to engineer the profile of this curve to have much longer breakdown times depending on the nature of the water environment in which we're dosing. So we can have products which break down more quickly if for short pipes and break down more slowly for longer pipes. As part of all the trials we do, the UK Environment Agency, which is the government agency that manages the open waters in the UK, require us to conduct monitoring of macroinvertebrate communities before and after dosing, and we've never had any measurable impacts on the biota of recipient waters. So we've had no impact on native bivalves, crustaceans, insect larvae, or any other biota, including fish and newts. What's also an unexpected bonus with our trials is that native unionid mussels seem to recognize BioBullets as a non-food because we've tailored our design so much towards the Dreissenid mussels. And so unionid mussels take the particles and never ingest them. Instead, they bind them in mucus and they throw them out on these long mucus chains as what we call pseudofeces. They've never gone through the gut. And so what we can see in this image is a close-up of one of these mucus threads loaded with rejected BioBullet particles by this native mussel. And this comes from some studies in North America with the USGS a number of years ago. And this is a black sandshell Legumia recta. But we've actually found that 3 genera of North American mussels respond in exactly the same way, and just the same with European mussels. Is it approved by regulators? Well, that's obviously an important question, and I'm pleased to say that it is. The products are endorsed by the UK Environment Agency for mussel control in the open environment. We're about to use it to control a new invasive bivalve that's just arrived into Europe. We have two formulations, SB1000 and SB2000, which are approved for use in UK drinking waters, which is perhaps the most tightly regulated environment there is. And these are criteria that determine approval in drinking waters across Europe. The products are fully manufactured and packaged under full ISO compliance, so they're ready to go with all the right paperwork. And we have, therefore, all the documentation needed to support approvals in new geographies and sectors, such as in North America. So how much is needed of our products, and what might a typical dosing regime look like? Well, we can tailor our service models to a particular utility and customer need. Our typical approach is to do an annual end-of-season dose for about 5 to 10 days. We use an approved service provider to deliver the product, and a typical dose is in the order of 1 to 2 milligrams per liter product. So this is the encapsulated product, not the active ingredient. So all you need is about 1 to 2 parts per million of product to achieve high mortalities. We've dosed in flows of up to 150,000 cubic meters per day, which is the equivalent of 122 acre-feet. And importantly, we have products which are effective against not just zebra mussels, but also quagga mussels. We've had 100% mortality through independent trials in the Netherlands. We've had 100% mortality against Asian clams in irrigation systems in Spain. We've had 100% mortality against Mytilopsis and marine mussels in South America. And we've also got some very encouraging results against other filter feeding organisms such as bryozoans and sea squirts, tunicates. So how can you see it in action? Well, what we're looking for is some opportunities for demonstrating our products in North America, either in water utilities, power plants, irrigation systems, or other systems that use fresh waters. So our next step is to identify trusted service providers. We've identified some that we're talking with at the moment, but also we're very keen to find people who are keen to work with us to demonstrate the products over there in North America. We are aware of the need to talk with regulatory authorities. As I say, we have the paperwork and have been through these processes in Europe. So we're confident that, that we can achieve approvals in North America. And we're looking also ultimately for licensing opportunities and partnerships. So I hope I've shown you that BioBullets offers an exciting and effective solution to management of mussels in North America. And I would really like to talk to people who are interested in working with us over there to make this an effective solution for you. Thank you very much.
Thank you, David. We have been collecting your questions as we've gone along here. So that concludes all of our presentations for today. So I'm going to move on to those questions that we've collected. As a reminder, you can use your questions pane of your GoToWebinar dashboard to submit your question or comment. And the first question I have is actually for Denise. Denise, are you still with us?
I am. Can you hear me?
Denise.
Hi.
The first question for you is, what buffer do you use?
We actually use just simply baking soda, and it seems to be effective and it's readily available. So it seems to be effective for our samples. We have a couple of locations where we need something a little bit stronger, but we do generally just use baking soda.
Thank you. The next question is for Jim. Jim, when you discussed using the technology for carp, have you noticed any issue with scavengers consuming dead carp or mussels and being harmed by the toxicant?
So far, there hasn't been any notice of that. That'll probably, if there is an impact, will be shown a little bit later on when there's some more extensive studies done in the pond and in the field.
And I have two more questions.
We also don't, sorry, we really don't anticipate that many side effects because antimycin does break down quite rapidly in the environment.
So I have two more questions for you. The first one is, do you have a hypothesis as to whether or not zebra and quagga mussels will consume the chemicals readily? And has there been past use of the chemical on both of those mussel types?
We do think that we'll be able to get those animals to consume the particles that we're working on. And there is a history of toxicity information with the niclosamide on Drusinid mussels. It is fairly toxic to those animals. And I think it has been used in some cases as just a control tool, not in an encapsulated form, but applied in Canada, if I'm not mistaken.
Thanks, Jim. And the next question for you, could the Asian carp control that you spoke about impact juvenile bass that filter feed? And were the bass in your test juveniles or adults?
They would have been larger animals that would not have been, early life stage stuff. So, is there a potential that they might grab a particle, and enough of them? Potentially, there would be a particle or an issue with that. I think that would need further investigation, and also, time of year of application could get around some of that as well.
And Denise, I'm going to go back to you with another question.
Okay.
What magnesium chloride concentration was most sensitive for the PCR detection?
We had two different concentrations for zebra mussel. It was right at 3. The standard method required 2. And for the quagga, she is now using 2 rather than 3, though we started off using 3.
Thank you. David, we have a number of questions for you, if you're ready.
Okay. Yep.
The first is, we had one participant ask if it was possible to share the U.S. patent number for BioBullets.
Yes, I don't have it to hand, but it should be readily searchable.
Okay.
But I can, otherwise please direct somebody directly to us and we'll share it with them.
Okay, we'll connect you after the webinar. And then, do you have EPA registration for the use of potassium chloride or other encapsulated toxins? And that would be US EPA. And if so, can you share the registration information or the current status of that process in the US and Canada?
We don't have the approvals at the moment. What we're hoping for is to find a partner who can, who's enthusiastic to work with us on conducting a demonstration in North America. And once those discussions are underway, then we will start the discussions with EPA.
Thank you. So we have a couple of questions related to this. So I'm going to try to address them all at once. But we have a number of questions related to the use of BioBullets in large lakes, large bodies of water such as the Great Lakes, and if you have any experience with using them or what you think about the potential for using them in large open lake systems. And if you have done any of that, what the results have been.
Sure. So the product is developed at the moment primarily to work in flow-through systems such as irrigation systems or raw water pipes in power plants or drinking water utilities. And that's because the mussels will experience continuously fresh products going down past them all the time. So open water dosing isn't something which we have looked at any real detail. But having said that, we have done some small-scale trials which have looked promising, and it requires a slightly different approach to dosing the products and a slightly different formulation to increase the period in which the products remain active. So we're about to do a trial in the open water in the UK, as I mentioned during my talk, against an invasive clam from North America, actually. So these things go both ways. And so we're looking. So we will have some results from trials hopefully this year on that. But very interested in discussing the opportunities of open water dosing. We have some other tricks up our sleeve which we might be able to use.
Thanks. So in relation to a related question, then we can assume that there's no successful eradication examples in open water bodies to date.
Not to date, although with this trial that's being championed by the UK Environment Agency, the target is very much to try and achieve an eradication in the open water. Certainly, the products that we produce allow us to control in large volumes of water. The product's well suited for large volumes of water. So there's certainly the potential. I don't think there's the potential of achieving massive kills in a Great Lake, for instance. I think those volumes are just too immense. But certainly in smaller water bodies, there is potential.
Thank you. And so we have a couple of questions related to more of the formulation and product itself. So the first is you mentioned two different formulations. Are they two different cores, or are they different coatings? And are the registrations for the coatings or the coatings and the cores?
Okay, so those two products carry different ingredients in the core. The coating material is the same in both. And actually, the formulation includes many different elements to achieve the target toxicity to the mussels. So obviously, if we're going to do any trials in North America, we would share the precise formulations with the regulators to achieve the green light from them.
Great. And then the next question is, do the BioBullets, are they able to encapsulate liquids as well as solids, or only solids?
The products I've spoken about carry solid active ingredients. We do have some processes where we can produce liquid active ingredients, but the products we're producing at commercial scale have solid active ingredients. It's probably worth also saying that the coating materials that we use are all food grade or feed grade. So we work with a company that produces these particles for a lot of food manufacturing processes. So they're very they're very environmentally safe formulations.
And then is there any information you can share about the average cost for BioBullets, either a cost per acre or cost per square feet, or any information you can provide in regards to that?
It's something I would very gladly share and discuss with anybody who's interested in taking a collaboration forward. We do have some rate cards, but it's difficult to give precise details in a generic form. But yes, by all means, if people are asking those sorts of questions, then we'd be very glad to chat with them offline about that.
Great. So I think that is all of the questions we've received for today. I'll give all three of our presenters one last chance if there's anything else you wanted to add or you forgot to say, or any other comments or you'd like to share. I'll give you an opportunity to do that now, and we'll just wait one more minute to see if any other questions come in. But Denise, Jim, or David, if there's anything you'd like to add, please feel free to do so now.
Thank you, Erica. I don't, yeah, me as well.
No, there's nothing from me. I think the questions that I got asked indicate that people understood what I was saying.
All right. Well, we haven't received any more in this last minute or so, so I'm going to go ahead and bring today's webinar to a close. Again, I want to thank all of our participants on the webinar today, as well as our presenters. As a reminder, we'll be posting the webinar recording on our collaborative website, along with additional materials, and can connect folks to our presenters today offline if that is helpful. If you have any additional follow-up questions or comments, please feel free to contact us at the Great Lakes Commission. And thank you again to everyone.